Welcome back. This week we get to talk about protein function, which is the follow-on to last week's discussion of protein structure. And I would say last week was all about pretty molecules. We saw beautiful structures, architectures that were fascinating, but I didn't tell you what makes them so special. I didn't tell you why it is that those structures allow proteins to do unique things. And proteins really are the superheroes of biology. These are the molecules that make it possible to change around whole solutions. And they make transformations possible that otherwise would be kinetically and thermodynamically inaccessible to the cell. So they're doing reactions, they're catalyzing reactions that otherwise, without these, biology would not be capable of taking place. Okay, so specifically this week, we're going to be talking about some pharmacology. We're going to be looking at dose-dependent response. We're going to look at non-covalent binding. And then, by analogy to non-covalent binding, we're going to make the leap to catalytic binding. And we're going to try to understand how enzymes work. I want to talk about how we're going to measure enzyme activity. We'll talk about how they're regulated. We'll talk about their mechanisms. And then we'll talk about mutagenesis and engineering. So these topics in here are going to give you the foundation that you need to make predictions about how enzymes work. And the overarching goal is for you at the end of this series, at the end of this 2 lectures, for you to be able to look at some reaction and then maybe not design the perfect enzyme, but make some predictions about how that enzyme might work. Okay, and let me give you an example of that. And this is an enzyme that we won't be talking about, but it's one that I want you to think about. This is actually a quote from the Iliad. And the quote is, "As the juice of the fig tree curdles milk and thickens it in a moment through though it is liquid, even so instantly did Paeon cure fierce Mars." This fascinates me. So in this reaction, you can take a branch off a fig tree, break it open, and actually this kind of milky liquid flows out. And you could drop that in a big bucket of milk. And what will happen is exactly what's described here in the Iliad. Now, our goal is to understand how an enzyme like this might work. If I tell you that the milk is getting solidified by a certain reaction, then you might be able to predict what the enzyme is, the enzyme mechanism is that makes that reaction possible. Okay, so that's our goal. A little bit of a mystery to set things up. Let me talk about some announcements first, and then we'll get into the meat of the discussion. So this week, please read Chapter 7, work the odd problems as usual. Our midterm is going to be a week from Tuesday. It will cover through Chapter, actually it's going to cover through Chapter 6, not 7, my bad. It will be comprehensive in the sense that there might be some concepts from the first 3 chapters, but it will focus largely on the more recent material. Okay? So when you're studying, what I'd like you to focus on are ideas and problems that are in the assigned homework, such as these odd problems in every chapter. I'd like you to focus on the problems that are discussed in discussion, and then I'll also post a sample midterm, which will give you an idea of the types of problems that I'm expecting you to know. Okay? So hopefully you're already starting to study for this, and that will be coming up pretty soon. Also coming up, abstracts for the proposal, the final proposal report, are due this Thursday at 11 a.m. We've already talked a little bit about the format of the abstract, but I've also released on the website more details about the proposal assignment, and I'd like to take a moment just to review those with you now. So very briefly, let's take a quick look. Okay. And on the website, let me go back. Here's the website, of course. Here's the proposal assignment. I have a very detailed description of the chemical biology proposal that you're going to be writing. In brief, what it tells you is that you need a simple idea. Okay, so in this first paragraph, it says, don't come up with something that's the next Manhattan Project. Don't, tell me if I get $1 billion, I could, do something like, solve toenail fungus or something, okay? Tell me something that I can do for, say, $100,000 or even less, $10,000, let's say. Those are the kind of proposals that attract attention. Clever ideas, things that people hadn't thought of. That shows brilliance. That shows creativity. The big ideas, sequencing 1,000 genomes. There are people who are doing that, and they're creative in their own rights. But that's not necessarily what this class is about. So simple, creative ideas, that interests me. Okay? And let me talk to you about some ground rules. You must choose a topic that will improve human health and well-being broadly. Okay, so there's lots of ways to do this. They could be things like improving the energy situation on this planet. If you have a new way of generating energy using enzymes, I would love to see it. And that would improve human well-being broadly. So the focus, though, of your proposal must be squarely qualified as chemical biology. If your proposal does not hit the topic of chemical biology, I will know in the abstract, and I'll give it back to you and tell you to change it largely. Okay? It's very important that it fits the definition of chemical biology. Your proposal must have a hypothesis or a very good reason not to be driven by testing a specific hypothesis. And then after that, you need to think about backup plans, creative variations, and further insight. So good proposals are a little bit like an onion. At the heart, you have some clever idea. You have something that if you could do this clever idea, it's going to change the world. But equally importantly, you have a lot of little backup plans and contingency ideas. So if the main idea doesn't work out, you have a bunch of backup ideas that are waiting in the wings that are going to rescue the whole thing and turn it around and make you famous. Okay? So that's the ultimate goal of a good proposal. And along those lines, you really should be having more than one idea. Okay? So a proposal has one great idea, and then there's a bunch of other little ideas that are kind of supporting it. Do not propose experiments that require human subjects or samples obtained from humans. This is important. I know many of you want to go to, I don't know, dermatology and medical school and become dermatologists later. And I've gotten proposals about picking scabs and things like that. Those do not interest me. That is not what this class is about. You know that's not what this class is about. So I will not accept any proposals that require you to collect samples from humans. Your proposal must include control experiments. We've discussed these. Positive and negative controls. We've discussed that. And then the next part is coming up with ideas. So how do you do this? The first thing you need to do is come up with a clever idea. Here is one very simple, formulaic way to be brilliantly creative for the rest of your career. Okay? If you learn this formula, you can be incredibly creative. Okay? And I'll be honest, this has always worked for me. All I do is I take a new technique, and then I simply apply this new technique to the problem that's already existing. Okay? And you too could do this. We've been talking all quarter about new techniques. You have new ways of screening libraries. We talked about RNA aptamers, for example. We talked about phage display. We're going to talk today about measuring enzyme activities. There are all kinds of neat new techniques that you could use. You simply apply those new techniques to an existing problem, and boom, you're creative. That's all it takes. That's all you have to do to be creative. Is you scan down the list of all the new techniques we've presented in this class, you scan down the list of problems, you take column A, and you take column B, one from A, one from B, you put them together, and again, boom, you're creative. That's all it will take for you to come up with a creative new idea. But it is essential for this idea to be creative, for it to be novel, and if it is not, I will return it to you ungraded. Okay? I'm fiercely defensive about creativity. This must be creative. Okay, now the other thing is, after you come up with this idea, you need to verify that in fact it's original. This idea can't be so outlandish as to be impossible, but on the other hand, if it's already been done, it doesn't count as creative, even if it's brilliant. What I usually do next after I come up with a creative idea is simply type it into PubMed, simply type it into Google, and see what else has been done in that area. If it turns out that someone else has done this idea that I thought was brilliant, Yahtzee. Yahtzee. I think that's fantastic. That tells me that my idea was brilliant enough that someone's willing to invest their own money in it. It doesn't bother me at all if I'm coming up with ideas that other people are willing to invest in and do. In fact, if anything, that tells me that I'm on the right track. And that should also tell you that you're on the right track. So don't panic. If it turns out that other people have come up with the idea before, that's perfectly acceptable. And it's actually kind of normal. And it's a good sign. It means you're on the right, you're going in the right direction. OK, so we talked a little bit about different ideas. Let me scroll down a little. OK, so here. After you finish screaming eureka about your idea, the real work begins. You have to dig into the literature, learn the field just a little bit, and know something about the area that you propose. Okay, so that's really the real work of this proposal. If you spend all of your time trying to think of the original idea, you're wasting time. Okay, 95% of the effort comes after you have the idea. So only give yourself a limited amount of time to think of the new idea, and then after that, start doing research and get that idea into shape. Don't spend a lot of time just cycling through ideas and you know, kicking yourself and saying, oh, it's not the world's greatest idea. Fine. You don't need the world's greatest idea for this assignment. What you need is an ability to argue successfully for that idea. That's what I'm grading you on. Okay? So along those lines, you know, OK, so yeah, so anyway, focus on that sort of thing. 95% of the work comes after you have the idea. OK, we've talked a little bit about the format of the abstract that's listed here. Here's some stuff about the format of the assignment. The assignment is going to be around 5 pages, not more than 10. I don't want to read it if it's longer than 10. No one wants to read it if it's longer than 10. Somewhere in there, there should be lots of figures. This is important. The level of detail should be sufficient for someone in this class to understand what you're proposing. You should be able to hand it to a random stranger in the class, just turn to the person on your right, and hand him or her the proposal, and then that person should get some idea of what's going on and should be able to judge it. That's the level of detail. You don't need to tell me about every experiment. You don't have to tell me where you're going to buy the materials and stuff like that. And most of all, you don't have to do the actual experiment. Okay, good news. Your Visa card is safe because I'll tell you, those experiments are expensive. And then finally, if you'd like to have your graded proposal returned to you with comments aplenty, then you have to give me in advance, attached to the assignment, a copy or a self-addressed stamped envelope. If there is no self-addressed stamped envelope, then I'm going to assume that you don't want any written comments back on your proposal. And that's quite all right. I just don't want to spend time writing comments if it turns out you're not going to pick it up. But if you want comments back, I will take the time to comment on your proposal. The TAs will take time to grade it and comment on it as well. And so you'll get back something that has some feedback to you. But I know not everyone wants that, so it's totally up to you. Last thought, it's important as usual that you turn it in through the turnitin.com website. This will be scanning for plagiarism. Bad news, I've already picked up plagiarism on the last assignment, which is disappointing to me. It happens every year, though. It drives me nuts. We've already talked about it. I'm not going to spend more time belaboring it. OK, now is the time I usually ask you for questions. If you have questions, don't hesitate to shoot me an email. Ask the TAs. Miriam, Krithika know a huge amount about this assignment. They can help you. OK, let's move on. So next, I want to talk to you about office hours. I have office hours this week, even though I'm not here on Tuesday. I will have office hours at the usual times and usual places. Thursday, immediately after lecture, and then Wednesday, 2:15 to 3:15 in my office. Miriam has her office hour on Friday, and Krithika has her office hour on Tuesday. Usual times, usual places. OK, that's it for the announcements. Let's get on with our regularly scheduled program. I want to talk to you about enzymes. Enzymes are truly remarkable. They are attractive to celebrities like these two. And they're also attractive to chemical biologists. And I would say they're attractive for the same reasons. They're attractive because they do transformations that would otherwise be inaccessible. Enzymes make possible the things that otherwise would be impossible, that would otherwise just take too long, or require too much energy. And we'll talk a little bit about how they do that. And I'm just, so entertained when I find, celebrity endorsement of my favorite topic. Here in the New York Times last year, actually on the same day I was delivering this lecture last year, February 22nd, 2012, enzymes try to grab the spotlight. And there are tons of enzymes that are found in papaya. They're called papain, and they're notable for digesting proteins. They're notable for digesting skin, for example. They're actually used as treatments for people who are undergoing therapy after bad burns, after bad high temperature burns, and it's a way of digesting away necrotic tissue. These guys have clearly some good facial structure here, so maybe they're onto something. Okay, let's talk next about how this topic ties in with what I told you about on last Thursday. Last Thursday, I was showing you how simple rules can dictate protein structure. And in a moment, we're going to then be applying these same rules to understand enzymes. Protein structure leads to protein function. So the shapes that the proteins were assuming on Thursday are what allow the proteins, these enzymes in this case, to actually acquire their unique function. And what I found so beguiling about this idea of conformational analysis is that the rules are so simple. We're talking about something as simple as just eclipsed versus staggered ethane, or gauche versus anti-butane. This fascinates me. This will keep me running to work for a really long time, because that's such a simple idea. These are such foundational concepts in modern chemical biology that I can explain to my grandmother. These are things that totally make sense, right? It makes sense to you that you would want to avoid electrons, which are all electrons banging into each other, right? Electrons hate banging into each other. It makes sense that things should try to spread it furthest apart. And if we think about things in that way, then such complex structures as the proteins that we're about to look at, their structures start to make sense as well. We talked about how, as a consequence of these simple rules, some amino acids can be found in specific types of secondary structure. And there are tables of this. And you could even make predictions about the secondary structure of proteins based on nothing more than the amino acid sequence. And surprisingly, these predictions turn out to be pretty good. They're about 80% accurate or so. And from there, people have been attempting to predict protein structure for a long time, and quite a bit of progress has been made in recent years towards that goal. We also discussed disulfides, which provide spot welds that hold together independent regions of protein structure that otherwise would sort of flop apart. And we also talked about how readily exchangeable these disulfides were to allow formation of new disulfides and exchange of one disulfide for another. The next topic we discussed was this hierarchy of protein structures, from primary structure to secondary structure to domains to tertiary structure to assemblies, etc. And so this helps us organize our thinking as these assemble into complex architectures. So we're going to be making reference to this as we start talking about enzymes. And then we ended with the concept that a relatively small number of protein domains are found very commonly in the human proteome. Now, the truth is, I didn't finish the discussion of this, so I need to pick up just very briefly with just a little bit more about protein structure, and then again, we're on to enzymes. So enzymes, last thoughts on protein structure. This is an example of an all-beta sheet protein called an immunoglobulin domain. These oftentimes assemble into long strings, and these are used in proteins like titin. Titin is found in muscles. Have you ever wondered why muscles are so strong? You've probably seen, for example, on the Olympics, you've seen Olympic weightlifters, you've seen them flexing these steel bars, right? They're lifting this stuff up, and the steel is flexing, and the titin in their muscle is hanging on. Well, what happens is there are long strings of these immunoglobulin domains that are lined up like beads on a string. So you have Ig domain, that's another word for immunoglobulin. So you have Ig, 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 and as you pull on the ends, one of these can unfold without snapping the whole muscle fiber. So the titin actually is holding things together. So an individual domain can unfold without breaking the entire protein chain. And that gives muscle, and that specifically gives titin found in muscle, some remarkable properties as a material. Okay, so on the left, this is the protein that's found in muscles, and I'm only showing you one immunoglobulin domain. Notice that the ends are 180 degrees apart. This then sets up these long strings of titin that can extend, up to the roof up here and then down through the floor down here. You can have many, many numbers, large numbers of these lined up. On the right, here's an example of an enzyme along the lines of the kind of thing I'm going to talk to you about today. This is a really remarkable enzyme. This is one of those enzymes that makes it possible for you to live on this planet. This is an enzyme called superoxide dismutase, and its active site is actually not where you expect it to be. I think when you look at these immunoglobulin domains, these are beta sandwiches. Your expectation is that the inside is this kind of deep cave. And that's not the case. Instead, this inside is chock full of side chains. This is full of stuff. It's only actually on the outside where the real action is taking place. Outside, over here somewhere, is where the active site of the superoxide dismutase is found. And incidentally, it's mutations in this enzyme that are responsible for diseases like Lou Gehrig's disease, which is a truly terrible disease. And so mutations to these kinds of proteins, these enzymes, have very, very serious medical consequences. Okay, last thought on immunoglobulin domains. They're named after the antibodies for which they're found in. And this is, again, one of these truly ubiquitous domains. Each one of these lobes over here is an immunoglobulin domain. And notice how these are just all strung together into an antibody. It should come as no surprise to those of you who were attending last week's lecture that the business end of antibodies, which are professional binding proteins, are found at the loops. Okay? So these antibodies are designed for binding to things. That's their role in life. They're professional binding proteins. And in order to get that kind of binding, the loops out here are exactly where you can pick up that kind of binding. Those loops are flexible. Remember, we talked about the low number of hydrogen bonds to the backbone. We talked about how the loops can accommodate many different shapes and sizes. That's what equips antibodies with the ability to recognize foreign attackers. Right? You cannot set up in advance an antibody against everything on the planet. These have to be just ready to pick up random things that you might encounter when you visit, I don't know, the taco wagon out here or something like that. So you have to be ready for that kind of thing, not knowing in advance what the shape is going to be. So having these flexible and molecular recognition versatile domain with these loops over here to accommodate diverse binding partners turns out to be key to understanding their activity. All right, another beta sheet protein, a good friend of mine, one I've published many papers either using or studying, is a protein called Steptavidin. And good news, we're going to be talking about this again in about 15 slides from now, so it gives me great pleasure to introduce you to the wonderful Steptavidin. Steptavidin is charged with binding to biotin. Again, this is an all beta sheet protein. Oh, there's a little tiny alpha helix, but it's largely all beta sheet. And it forms these wonderful little beta barrels. At one end of the beta barrel, this small molecule called biotin sits. And we'll look at the structure of biotin in greater detail in a moment. This is an example of a small molecule. The molecular weight of biotin is 254 grams per mole. It's tiny, tiny, tiny. And biotin is trapped by the quaternary structure of Steptavidin. And this is an important concept. If we look here at, say, the secondary structure, the tertiary structure of Steptavidin, your expectation is that biotin is not going to be very firmly held in this beta barrel, right? I mean, look at all the space. You can imagine the biotin just floating away and coming out of the Steptavidin without very much trouble. Okay, so tertiary structure doesn't begin to hint at the extraordinary abilities of this molecule to grab on to biotin. And here's what I mean. Okay, so now this is the quaternary structure of Steptavidin. And Steptavidin consists of it consists of a homotetramer of 4 streptavidins that are non-covalently joined together to trap biotin. And notice there are 4 biotins bound here. Now, what's happening is the alpha helix from a neighboring streptavidin is sticking down over the top of biotin. I've highlighted that for you in black over here. And you could see it's actually forming a trap door to slam down over the top of the biotin and prevent the biotin from floating away. That turns out to be key to its activity. So streptavidin evolved to bind biotin with astoundingly high affinity. And we'll talk about the exact number in a moment, but it is really extraordinarily high. And it evolved to bind this cofactor called biotin as a way of killing any bacteria that happen to be present. So for example, a related protein called avidin is found in egg whites. Okay, so egg whites have a high concentration of avidin, and that means that if any bacteria try to colonize the egg, I'm talking about hen eggs here, if any of those bacteria try to get in there and go to town and eat all the juicy richness of a wonderful egg white, they're going to die because their biotin will get sucked up by the streptavidin and then trapped almost permanently. And that turns out to have fatal consequences for the bacteria. Turns out that it also can have fairly fatal consequences for humans. If you live on a diet of nothing but egg whites, your biotin will also get pulled out of your body. And this is a kind of an astonishing fact because it turns out it doesn't take a lot of biotin for you as a human to survive. Biotin is an essential cofactor in the synthesis of lipids. And, but however, there are eccentric people out there who constantly do experiments like this. And when they show up in hospitals after eating a diet of nothing but egg whites, the physicians tend to be totally baffled because they're not used to seeing such bizarre symptoms. And so there was a case, I'll send it around, I think this is in the New England Journal of Medicine, of this English guy who showed up and was living on a diet of nothing but tea and egg whites. And he had all this like bleeding out of his gums and his pores, and anyway, he was falling apart basically. And the astonishing thing is it would take just a few micrograms a day of biotin for him to be totally healthy. But there's such a high level of avidin and its homolog again to streptavidin present in egg whites that it was actually leaching all of the biotin out of his body. Okay, pretty extraordinary biochemistry, pretty extraordinary molecular recognition. It absolutely fascinates me to understand how this works better. It's something I've spent a lot of time thinking about. Okay, and yet another example of a very common protein fold that's found in the human proteome. The WD proteins consists of these beautiful propeller-like assemblies where there's actually 7 of these little triangles. They look kind of like slices of a pizza that come together to form these large assemblies. These act as scaffolds to organize big machines found inside your cells. So this, each one of these faces over here might bind to a different protein and bring it together like it's an assembly line for putting together really complex things inside your cell. Absolutely fascinating stuff. Something else that's truly bizarre. What's up with the 7-fold symmetry? I think I'm ready for 4-fold symmetry, but we humans don't like to think in 7-fold symmetry. So I'll just leave that as something for you to puzzle over. Also, another very common, and I'm switching gears, we're going down the chart of the most common protein structures found in humans. Collagen is a very common protein structure. It's actually an unusual 3-stranded coil. It differs from the alpha-helical coils that we saw. The twist is different. And it's a little hard to see, but there's actually 3 different colors. There's a green color, a purple, and a blue color. So there's 3 different colors here. There are these 3 strands that are winding around each other. And this makes a very strong framework for collagen. Collagen is another one of these ubiquitous structural proteins found in the body. Notably, this protein requires a post-translational modification, introducing a hydroxide into proline residues, and that has the effect of setting up one particular preferred structure, a particular twist in the collagen triple helix. Without this hydroxide, the protein is unable to assume that conformation. All right, let's switch gears. I want to talk about GPCRs. This is the class of proteins that makes it possible for you to see me, that makes it possible for you to smell, for you to taste, pretty much any of your senses. totally dependent upon this class of proteins. So I think we should take a moment and be grateful for their existence. How do these things work? Okay, so this is a really, how is it that you're going to sense a photon? How can you actually, respond to light, which is, you can't bind light, right? So how do these things work? All right, so in short, these sensing proteins, again, they're called G-protein coupled receptors, or GPCRs, are all alpha helical. And notice that there's they each have 7 alpha helices. These 7 alpha helices transit plasma membranes in the cell. So going through here is the plasma membrane. So there's an outside, an exterior, and there's an interior facing the cytoplasm. Exterior is facing the extracellular milieu. And these change conformation upon binding to things. In the case of a photon, the GPCR responds by having a isomerization of a carbon-carbon double bond. So the photon hits, it isomerizes this carbon-carbon double bond, flips it from one configuration to another configuration, from trans to cis and back. And in doing so, that rearranges the conformation of these residues down here that are found on the inside of the cell. Okay, last thought. Notice that this is a coiled coil, and if you look carefully at this, it is a left-handed coiled coil. Almost all of the coiled coils found in nature are left-handed. Notice that your hand can trace this out in a left-handed manner, doesn't want to trace it out if it's right-handed. Okay, so these are very common because alpha helices in general are hydrophobic secondary structure. They fit nicely into membranes, and this property makes them very useful for sensing what's happening outside the cell and communicating it to the inside of the cell. Next one, here's another example of an I'm going to talk very briefly about alpha-beta proteins. This is an enzyme that we'll talk more about very shortly. This is a barrel-like protein. Here's the barrel in the center. This is the active site. So you can mix and match alpha helices and beta sheets. I'm showing you this because I didn't want to give you the impression that secondary structures never mix. In fact, they're very commonly mixed together. Repeat proteins. So repeat proteins are another very common assembly of proteins. This is an example of an anchorin repeat protein. Notice that it has 2 sides. The concave side has these loops, and wouldn't you know it, the loops turn out to be the key to this binding activity. This is a protein that's used, it's very versatile. It's used in many different contexts. And what happens is these loops are mutated to give a particular binding property that's then useful for the cell. Its corollary is a leucine-rich repeat, which now has loops on the convex side. Okay, so the anchorin repeats had loops on the concave side. This one has loops on the convex side. And these loops then can grab on to the binding partners. Okay, analogous to what we saw with the antibodies. Okay, so these two are very versatile structures that can be evolved through organisms pretty readily, and that equips organisms with new binding activities, which in turn can be used to respond to environmental changes, etc. All right, last structure that I want to talk to you about, peptide binding domains. This is an example of the SH2 domain. This tiny little domain shown here in yellow binds phosphotyrosine proteins, so proteins that have been post-translationally modified using a class of enzymes called kinases, which we'll talk about in a moment, bind to this SH2 domain and fit into very deep pockets. Okay, so there's a deep pocket down here. These evolve basically to have specificity, to bind to a particular sequence, so they're not binding to every phosphotyrosine, they're picking out specific binding partners. Another peptide binding domain of note are SH3 domains. These bind polyproline helices. This is the I+3 helix, the 310 helix that we saw during Thursday's lecture. And this binding pocket is a very shallow one. It almost looks like the peptide is resting on the top of this SH3 domain. It's like a butterfly, kind of alighting on the top of the SH3 domain. And notice how delicately folded it is into this threefold symmetry helix, right? So you can actually, if we look down one axis of this helix, you can see how it's forming this polyproline I+3 helix. Okay, it's hard for me to talk about this without having a few favorites. And I know as a chemical biologist, I shouldn't have favorite molecules, but I do. This is one that I spent years of my life thinking about in my wasted youth. I was interested in these MHC receptors for reasons that are too bizarre to explain right now. But suffice it to say, these are the receptors that your immune system relies on to let it know when the redcoats are coming. These are the receptors that raise the alarm when foreign invaders are trying to take over your physiology. And the way this works is a small percentage of peptides that are synthesized by the cell are digested and then displayed out on the surface of the protein, just like little flags. And the idea is that if a virus has taken over the cell, little flags of virus will appear outside on the surface of the cell, and the immune system then knows, oh no, that cell has been infected with viruses. I better kill the cell. I better mount a strong response. This is a very effective way of alerting the immune system. Okay, here's some old friends as well. Notice down here, do you recognize that domain? Does that look familiar? Yes. That is the same domain we saw a few slides ago. This is the beta sandwich immunoglobulin domain. And here it is making a cameo in a slightly different but equally important role. Here it is actually lofting the peptide out off the surface of the cell. Down here is the surface of the cell. Here's the scaffold of this immunoglobulin domain, kind of holding it above the surface of the cell, making it a little easier for the flag to be seen by passing T cells, especially cytotoxic T cells that take an interest in these things and then can go into action and kill the cell if necessary. All right, one last example of this. The last example is a slightly different variant of these MHC receptors. In this case, this is one called class 2. The one on the previous slide was called class 1. The details, not so important. But this guy actually displays peptides, not that are being synthesized by the cell necessarily, but peptides that are being engulfed by the cell. So what the cell is kind of randomly taking up extracellular material. And so again, this gives the immune system a different look. So the MHC class 1 tells about it reports on what's happening inside the cell. The class 2 reports on what's happening outside the cell. And notice that the structure of this peptide, when viewed down the same axis that we looked at earlier from SH3, it again starts to bear some hallmarks that we've seen before. Notice that it also has that sort of threefold, it kind of looks like a triangle type of geometry. And yes, this is also assuming a polyproline type helix, analogous to the I+3 helices that we saw earlier in this class. Okay, so hopefully some of the concepts that we saw earlier are finally coming into play. And what I want you to do is I want you to first have an aesthetic appreciation of these things. These are beautiful. Okay, I like to think of this one as like a hot dog in a hot dog bun. I mean, look at this thing. It's so juicy I could eat it. But equally importantly, it has these immunoglobulin domains, again, that have the wonderful function of lifting it off the cell surface. At the same time, it's presenting lots of surface area out here, so the cell can recognize whether or not this flag belongs to self or non-self. It can even interrogate this one receptor and determine whether or not the original cells are from self versus non-self. And this is really one of the challenges for organ transplantation, is to deal with this class of proteins where where different humans have different MHC receptors. And this is one way that the immune system keys in on organs that have been transplanted and knows to kill them. Okay, now obviously I can talk about this particular topic for hours, but we don't have hours. So we'll have to move on. All right, I want to finish our discussion of protein structure by talking about higher order assemblies. And I guess our best example of this is a structure that I introduced to you earlier, collagen. Collagen, again, plays this key role of structurally strengthening bones, of joints, of doing all kinds of important things. This is, yet again, one of those proteins that makes it possible for the weightlifter to hoist, do a deadlift over his head and have the bar flex. I've always wanted to do that, but I don't think it's going to happen. But just to see the bar flexing is, a thing of beauty, right? How is that possible? Okay, so obviously these things are really strong. Collagen is remarkably strong. It also is assembled into an ordered structure where that ordered structure supports each of its constituent fibers. And I guess the best example of this that you'd be familiar with is something like a rope. Right? The individual strands of the rope, of a fibery rope, not so strong, but when you wind them together and they're all supporting each other, suddenly you get something really strong that can anchor an aircraft carrier to a dock or something like that. All right, so here's the way collagen works. You have to control its assembly in a way so that it doesn't kind of prematurely wind itself up and then get all tangled. Okay, so what happens is the triple helix is formed with some caps on the end. And these caps on the end remain for quite a while. So the end cap pieces are then brought together, and then the whole assembly with the cap still in place is secreted outside the cell. And where these red arrows are, these caps are then snipped off using proteases, the scissors of the cell. And that gives you formation of fibrils. Okay, so without that, this does not happen. Okay, so without that, you get this kind of sticky, tangled up mess. But what ends up happening instead is a very detail-oriented assembly where each step in the process is carefully controlled, and that's essential to making structures that are really strong. Okay, now I want to move on. I want to talk to you next about enzymes. We've seen protein structure, enough about protein structure. Let's talk about what they're good for. Okay, obviously they're good for strength and structure. I want to talk about catalysis next. Okay, so in order to talk about catalysis, I have to introduce you to some measurements of strength of binding, of catalytic efficiency. And so the first thing I'm going to have to do is define a few equilibrium constants for you. The first of these is used to describe the strength of a non-covalent receptor-ligand interaction, where the receptor is indicated in R and the ligand is this little sphere indicated in L. Now, if you have a bunch of receptors on the surface of the cell that want to bind to ligand, the ligand is going to hop on, it's going to hop off, it's going to hop back on, it's going to hop back off. So we need some way of describing the occupancy. How many of the receptors are actually bound to the ligand? How many of the ligands are free in solution? So the way chemists do this is using equilibrium constants. These equilibrium constants are kind of special, so they get a special name, but they're more or less the KEQ that you learned back in Chem 1. So here's the way this works. We can describe some receptor-ligand interaction that's formed as having a dissociation constant in which the receptor and the ligand dissociate from each other. And that dissociation constant, Kd, is equal to concentration of receptor times concentration of ligand divided by the concentration of the complete receptor-ligand interaction. The inverse of the dissociation constant is the association constant, abbreviated Ka. Okay? But again, these are just fancy equilibrium constants. However, they tell us quite a bit about the strength of an interaction, and I'm going to be referring to them. One thing you need to know is that a lower Kd, a lower dissociation constant, means a stronger interaction. And we're going to stick with Kds. Okay? Everyone out in the pharmaceutical world, in the biochemical world, discusses things in terms largely of Kds, Kas. You basically, every time I hear someone give me a Ka, I mentally take the inverse of that number and then think about it in terms of Kd. It's just a convention, okay? But what matters is that a lower Kd means a stronger, stronger interaction. That means more of the ligand is bound here, right? So you have more of the complex formed, bigger number down here, lower Kd. let's take a look at some of these. Okay, I've been talking to you a little bit earlier about organ transplant and rejection. So after people receive transplanted livers, they're given a class of drugs called immunosuppressants that suppress the immune system. And we know quite a bit about receptor-ligand interactions through classic studies done by Stuart Schreiber and others that looked at how these immunosuppressants work. Here are two examples of immunosuppressants. On the left is a small molecule called FK506, and on the right is a small molecule called rapamycin. They both work by targeting a binding protein that the Schreiber laboratory named FKBP for FK506 binding protein. And here it is neatly fit into this receptor, which is FKBP. So the ligand is the immunosuppressant drug. The receptor is the FKBP. And notice that this is finding a really deep binding pocket to bind to. Let's zoom in, okay? Let's take a closer look. So imagine now that we can zoom in just looking at the green ligand. What we would see is something like this, where in blue, this is the region of the small molecule that's bound by FKBP. Okay, so again, on the left is FK506, on the right is rapamycin. Notice some similarities here. Okay, notice that in blue, these largely have the same structures. Okay, that's not a coincidence. That same structure helps orient the molecule and make it so that this half of the molecule can very readily bind to FKBP. Notice that the part that's not shaded, these two are wildly divergent. Okay, molecule on the left, the part that's unshaded looks completely different than the thing on the right. Okay, and that's not too much of a surprise either, because it turns out that these two molecules affect different pathways in T cells to suppress the immune system. And these ligands act as sort of like the meat in a molecular sandwich, and they recruit two different top layers of bread. This one over here recruits a different protein than this one over here. However, both of these molecules bind to FKBP with very high affinity. And the way we know this is high affinity is we refer to their KD, the dissociation constant, as having a sub-nanomolar KD range. That's really good. That's really, really tight binding. And it turns out that most of the pharmaceuticals that are approved tend to have affinities for their targets in this kind of very low KD range. Why that is will be apparent to you in a few slides. Okay? Okay, so this is one way of describing non-covalent interactions. I next have to tell you how we're going to be describing speeds of reactions, the kinetics that make reactions possible. Two kinds of reactions that we're going to be seeing in this class. Kind number one are unimolecular reactions. These are reactions where you have some reactant and it goes through a transformation and that's it. Okay, there's no other species that's implicated in the reaction mechanism. That's it. It's just this one tetrahedral intermediate falling apart, collapse of the tetrahedral intermediate. The rate of unimolecular reactions equals some rate constant, little tiny k, times the concentration of the starting material. Okay, this k over here, I'm emphasizing tiny k for a reason. The little k indicates rate constants. That's totally different than the equilibrium constants I was showing you on the previous slide. Never the twain shall meet. They're two totally different things. It drives me crazy, though, that they're both symbolized by k. And there's nothing I can do about that. Okay, we're stuck with that. It's old-timey nomenclature. All right, the next one. We're also going to see bimolecular reactions. These are reactions that have two reactants that are colliding with each other, and in that collision, resulting in formation of a new product. The rates of these bimolecular reactions are going to be equal to some little tiny k, the rate constant, times the concentration of reactant 1, in this case hydroxide, times the concentration of reactant 2, which we're calling Y in this case. Make sense? Okay, I expect that this has been a review. This is something you've seen before. In biology, though, these rates vary enormously. Okay, check this out. this rate, constant K1, ranges from 10 to the 13th per second to 10 to the minus 7 per second. That's 20 orders of magnitude difference in speeds. Okay, this is at the wild, fast end of the scale. These are things that are a total blur. And at the super slow end of the scale, these are things with geological times that are so slow, they simply don't even matter in biology without some sort of catalyst to speed it up. Okay, so these are the parameters we're going to use. Let's now think about kinetics first of non-covalent interactions, and then we'll talk about enzymes. Okay, so for a non-covalent interaction, you can imagine the ligands hopping off of the receptor. When that happens, there will be some speed of this that will have a rate constant of little k off. Similarly, ligands can hop on to the receptor. And again, there's a rate constant, little k on. Naturally, if this is at equilibrium, then you can actually, you can work out that the Kd equals the ratio of the K off to the K on. Okay? Everyone still with me? Again, this is at equilibrium where the, let's see. Okay. Everyone still with me? Okay, good. Here's the thing. The typical rates of binding are, again, wildly different. And this is a table from the book, table 6.1. I want you to take a moment to just gaze at the truly awe-inspiring nature of this differences in speed here. Okay, so let's just take a moment to appreciate this. What I'm showing you is a series of different receptors, and over here, a series of different ligands. At the top, these are small ligands. These are small molecules like biotin that we saw earlier. And at the bottom, these are large ligands. Okay, now check this out. This is really cool. Notice that the on rates for all the small ligands, roughly the same. Very, very little difference. They're all right in that 10 to the 8th range. And hey, guess what? 10 to the 8th is kind of near the speed limit. The speed limit for zooming through the cell is going to be somewhere around 10 to the 9th per molar per second. Okay, that 10 to the 9th is a physical constant. You can't bounce through water any faster than that. And so these small molecules are zooming along about as fast as they possibly can to fit into their receptors. But check this out. Off rates. These off rates vary enormously. They range from, say, 9 over here up to 100,000 over here. And down here, and the big things, huge changes in off rates as well. Okay, so what this tells us is if you are trying to design the perfect pharmaceutical, the perfect therapeutic to treat, I don't know, muscular dystrophy or something, you want to spend a lot of time thinking about off rates. Off rates are where the big money is when it comes to therapeutics, when it comes to pharmaceuticals. They all have roughly the same on rates. What differs is off rates. And those off rates are how you can determine whether or not a pharmaceutical can be given at a low dose versus a high dose. But I'm getting ahead of myself. We haven't talked about dosing yet. Okay, let's talk about the large ligands. Large ligands have enormously variant on rates. There's too many zeros here to count. And then over here, enormously variable off rates. This should make sense to us just intuitively because large molecules aren't going to be able to zip through the cell as quickly as small molecules, right? They're going to get, sidetracked. They're going to try to bind to other things. They're going to, their diffusion rates are going to be slower, for example. Okay, now let's put it all into effect. Okay, let's put everything we've seen so far into one summary that tells us what it is that we care about in terms of treating patients. Okay, so the way it works is what we want to do is we want to have some biological response. Okay, if our goal is to cure patients of, say, toenail fungus, then our biological response is going to be, what percentage of their toenails are clear from the fungus. Here's the way this works. On the y-axis, this is the percent biological effect. That biological effect results from ligands binding to some receptor. Okay, we've seen, for example, antibiotics that are binding to the ribosome In that case, the ribosome would be your biological receptor, and the biological effect would be death of the cell. That would be killing the bacteria. Okay? And so in general, we see sigmoidal biological responses when we look at receptor ligand binding. Okay? When it's graphed as log of the concentration of drug along the x-axis and % biological response at the top. At the very top, at 100% biological response, this will take a very high concentration of drug. Okay? Notice that the numbers are bigger on this side and then smaller over here. This is 10 to the minus 9 over here, 10 to the minus 1 over here, in concentration in molar. I realize this, the axis is a little confusing. Bear with me. There's a reason we did it that way. Okay, so up here at a really high concentration, 100% biological effect. Okay, but maybe at that concentration, you end up with a drug that has to be given with pills that are like, the size of, erasers or something, and no one likes to swallow things that are really big. So we compromise. Instead, what we want is we want to have a 90% receptor occupancy in vivo to see some sort of effect. Okay, that's our goal. So we're going to be measuring biological potency through these dose-responsive effect. And the major goal of pharmacology is to get up here into this 90% receptor occupancy where you get 90% biological effect, greater than 90% biological effect. Now, typically, the numbers up here, obviously we're approaching an asymptote, so things are, can go over a really long time up here. So instead, we describe biological potency in terms of an effective concentration for 50% effect. That 50% effect takes place right here at the point of inflection for the sigmoidal dose-response curve. And so we can compare two different drugs just by comparing the EC50, where the more potent drug will be the one that gets the same 50% response, but at a lower concentration. Right? It means then that the patient can be treated with a lower dosage to get the same effect and the same benefit. Okay, so I think it's worth us taking a moment to talk about this because like 90% of the students in this class are going to be spending the rest of their lives battling with this sigmoidal curve and trying to get up here and sometimes being down here. Okay, let's talk about how you measure biological response. Oftentimes in chem bio laboratories, this is measured using a ELISA, an enzyme-linked immunosorbent assay. And before I can tell you a little bit about how that assay works, I need to tell you about some reactions that are catalyzed by enzymes. There are two enzymes that are kind of the workhorses for chem bio laboratories. One of these is called peroxidase, other one's called phosphatases. These are reliable enzymes that will catalyze reactions that lead to turnover of dye molecules. So here are two molecules up here. This is these two aromatic molecules. These guys up here are clear. Okay, so if you made a solution of these guys, it would look more or less clear. It might look a little yellow, but more or less clear. However, after the enzymes, these two enzymes catalyze these reactions, what ends up happening is you get a dye that has a deep color, a very strong color. This one forms a dark black, actually more brownish color, very dark color. This one over here forms a bright yellow color. So both of these give us reliable indicators that we can use to follow how much activity is taking place at a certain dosage. So let me show you how this works. What we do is we use plates, and I think I've talked about those before. They're ELISA. They're called ELISA plates and colloquially in the lab. And these plates have 96 wells on them. Okay, so that's over here. And each one of these wells can be coated so that the surface is coated with the receptor. Okay, so here's the receptor down here. The problem is if the surface likes to bind receptors, it'll also bind to the ligand, and we don't like that kind of thing. So what we do instead is add a blocking agent, typically something like dried milk, non-fat milk. Not dry, it's solubilized, and so we take non-fat milk and coat any other place on the surface of this well that otherwise might start to bind non-specifically to ligand. We then add the ligand. Stuff binds, we wash away the non-binders, and then add an antibody against the ligand. So wherever the ligand is bound, we're going to get an antibody stuck to it. This antibody is a special antibody. Unlike the antibodies I showed earlier, this one happens to be covalently tethered to the enzyme that I showed on the previous slide. That enzyme is peroxidase, and so this means when you add the dye, this peroxidase goes to town and turns over the dye in this well, and then you can look at a large number of wells to get a dose-response curve. Okay, this works really well. This is totally robust. You can use this in your proposals. It works great. Okay, let's talk next about other receptor-ligand interactions. Okay, so I've been showing you ELISAs as an example of catalytic receptor-ligand interactions. I want to get back to streptavidin. Okay, so streptavidin has this remarkable half-life of 200 days. Recall that this is the protein that is found, an analog is found in egg whites, and if you live on a diet of egg whites, eventually you're leaching all of the biotin out of your body. This half-life over here of 200 days starts to make sense, starts to explain why it is that you can actually leach all of the biotin out of your body. Its KD happens to be an astonishing 10 to the minus 15th molar, or picomolar. Subpicomolar, really. Femtomolar would be 10 to the minus 15th. So subpicomolar KD. That's extraordinary, because earlier I showed you rapamycin and FK506, and I said that nanomolar affinity was really high. That's a really great binding partner. In this case, this is one that's a million times better, and it's really strong. Okay. Now, chemical biologists have learned to use this for all kinds of assays. One thing we do pretty often is attach biotin covalently to small molecules and use this kind of like lures for fishing. Okay, so you all know about fishing, right? So the way this works, you have a line, you throw the line in the water. At the end of the line is a hook. The fish are smart, okay? The fish aren't going to eat some random hook. So instead, we'll put some sort of lure on the hook that's then going to bring the fish up to the hook. Okay, and classically, I guess this was worms. I don't fish with worms. I fish with flies. But I also like to fish with small molecules. And when I do, I always use biotin. Okay, so here's the way this works. So here is the biotin. It's now covalently tethered to some small molecule. This becomes the lure. Okay, so this side over here is going to track proteins from the cell, and then this side over here is going to be attached, is going to act as a handle. So that's the part that you grab onto and hold. And where this is really useful is if you have some new small molecule and you don't know what it binds to in the cell. This happens to us all the time. We'll have some molecule that we pulled out of, I don't know, a fruit fly screen, where we're looking for molecules that make fruit flies less drunk or something like that. And we want to know, how does that work? Okay, so the way you would do this is you'd have some linker and then biotin, and then you basically go fishing and hope for the best. A quick word about the linker. The linker matters a great deal because you remember earlier I told you about the trapdoor and how the neighboring streptavidin subunit slips over the 1-beta barrel and traps it. Without this linker, it's very hard for these molecules to be strongly held by biotin. The linker allows the trapdoor to close all the way and get wedged tightly shut. Without this, then some molecule like this that has this ring system nearby would basically get into the trapdoor and block it and make it harder for it to close all the way. So the linkers actually matter quite a bit. Okay, in practice, here's what it looks like. In practice, we have these columns that we flow cell lysates over. And what we're looking for is we're looking for molecules that will stick to the column that are binding to not to biotin, but to the product, the target that's then tethered to the biotin. Okay, so this is the way we go fishing. The handle is over here, that's the biotin. It's grabbed onto by streptavidin, and then the lure is hanging out, and you send through junk from the cell, just cells that are chopped apart, and you hope that stuff sticks. Other molecules that don't bind to the target flow through and don't stick. Okay, and again, this works great. Okay, so everything you know about KDs are now being applied. Okay, so now what I've shown you is I've shown you an example of a really strong receptor-ligand interaction. I've shown you how to measure biological effect. Let's now zoom in and start talking about catalytic receptors. This is a great example of a non-catalytic receptor. Enzymes are basically catalytic receptors. Everything that we've been discussing in terms of dissociation constants, in terms of binding, in terms of molecular architectures, all of that comes into play when we discuss enzymes. So what do we talk about when we talk about enzymes? We like to talk about these in terms of a few simple parameters. And again, these are parameters that are familiar to us from our earlier discussion. So earlier I talked about receptor-ligand interactions having K-ons and K-offs. Similarly, enzyme-substrate interactions are going to have K-ons and K-offs. They can either form the complex, the enzyme-substrate complex, sometimes called the Michaelis-Menten complex, or they decide not to. And the one little twist here is the fact that the enzyme is going to catalyze transformation of the substrate to some product. Okay, substrate is a fancy word for starting material. Okay, and what is happening here is we're going to form this intermediate complex, and this intermediate complex is going to very quickly collapse. If it collapses to the left, if it collapses to the right, then it is going to then form a product. And this product will, you hope, quickly dissociate. If it collapses to the left, then the substrate diffuses away, and this goes through an off rate. Okay, quick word about this. In this case, I'm showing that the enzyme product, the product then has to dissociate from the enzyme. If that doesn't happen, then the enzyme is trapped and inhibited in this state. And guess what? That actually happens. And in fact, it's one of the reasons why oftentimes this last step can be a rate determining step for enzymes that you would not expect this to be a problem for. All right, let's zoom in and take a look at the reaction coordinate diagram for a enzyme catalyzed reaction. I will tell you in advance that this is grossly oversimplified. The real reaction coordinate diagram is complicated and messy, and I'm going to show you the theoretical idealized version first. Okay, so enzymes work by stabilizing and thus lowering the energy of the transition states. Doing that accelerates the reaction. Okay, so here's a typical reaction on the y-axis. This is the change in energy, delta G. Higher on this axis means less stable. Lower on the axis means more stable. And as you know, higher means takes more energy, which means then in turn it's slower. On the x-axis, this is oftentimes called the reaction coordinate. It's nothing more than the conversion going, or the pathway between enzyme plus substrate going all the way to enzyme plus product over here. Okay, and here it is going through a couple of different intermediates, enzyme substrate intermediate, enzyme product intermediate, and then also a transition state. Okay, so in order, let's talk about the uncatalyzed reaction first. Uncatalyzed reaction in blue, substrate starts off over here, gets converted to product. That takes a lot of energy. Okay, the enzyme in this case is uninvolved. That's why it has a plus sign. It's kind of hanging around as a spectator. Let's just imagine that would happen. So if that happens, there's this activation energy here, the difference in energy between the transition state and the starting material is very high. We can actually derive the speed of the reaction from knowing that energy, right? And we know a bigger energy over here, higher activation energy, means a slower reaction. And oftentimes these reactions are too slow to be biologically useful. If we relied upon spontaneous reactions taking place, you wouldn't be a human, okay? You would just, it wouldn't be possible for biology to take place. Instead, what happens is enzymes stabilize, bind to the substrate, lower the energy of the substrate complex, and in turn, most importantly, lower the energy of this transition state. The red arrow here is exactly how much lower the energy is. The bigger that arrow is, the better the enzyme, right? Because that's how much, how greatly improved it is. Similarly, the enzyme then binds to the product and then eventually dissociates from the product. The product dissociates. Okay, let's take a look at an example. The example is first a unimolecular reaction. I realize this is kind of a hairy example. bear with me, it's worth it. So in this case, what's nice about this example is this reaction is a concerted reaction, meaning it goes smoothly in one fell swoop. There's one, step in this reaction mechanism. This is a paracyclic reaction, and it involves the transformation of charismate on the left to prephenate on the right. And again, the enzyme is called charismate mutase. By the way, this is a key step in the shikimic acid pathway, which all the plants on this planet rely on to produce phenylalanine and the aromatic amino acids. This is one of those things that we humans could not exist on the planet without. We cannot do this reaction. Plants and microorganisms are the only organisms that have the necessary enzyme to catalyze this reaction, and for that matter, the only organisms that have this shikimic acid pathway, which is why indirectly you must eat plant material to survive. Okay, so check out this reaction mechanism. This is a beaut. This is awe-inspiring. Okay, so in this reaction mechanism, electrons are going to bounce down here, bounce, bounce, and bounce some more, all the way up to here, producing a new carbon-carbon bond between this carbon over here and this carbon. These guys are going to join together to produce this new carbon-carbon bond that has the wedge coming out towards us. Okay, so in order for this to happen, this part of the reaction mechanism has to be sticking out towards us. And this whole thing is just going to swing over the top. And all 6 electrons are going to be flying at once, fists of fury style, to give you, in a neat swoop, this new carbon-carbon bond while breaking apart this old carbon-oxygen bond and breaking a carbon-carbon double bond to form a new carbon-carbon double bond. This is really spectacular stuff. This is the stuff of just, graceful electron choreography in motion. Okay, let's take a closer look and see exactly how this works. Oh, and by the way, I think if you try to do this reaction in lab, you can get it to work, but it's going to be slow. It's going to be a dog. So check this out. This is the way the enzymes do it. This is the way they make this happen. What they do is they grab on to the handle up here and force it into some semblance of the necessary transition state. And in doing so, they're going to lower the activation energy of this reaction. Okay, so here's what it looks like bound, or actually I'll tell you in a moment what this is. This is kind of like the transition state bound to the enzyme. And in practice, the transition state looks like this. Here in dashed lines are the bonds that are being made and broken. And in order to get this reaction to take place, the enzyme has to grab on to this thing over here and basically force it over the top, so that this carbon and this carbon are next to each other to form a new bond. If that doesn't happen, then no reaction takes place. And so what the enzyme is doing is just grabbing on and forcing this thing into the right configuration to allow the reaction to take place. Beauty. It's a true thing of beauty. Okay, now here's the way we know that that's how that happens. This is classic work done by my advisor from my undergraduate days at UC Berkeley, Paul Bartlett. And the Bartlett laboratory synthesized an inhibitor of this reaction that looks like this. It kind of mimics this transition state. And guess what? This mimics the transition state so well that it sticks in there and just plugs it up. It binds really well to this enzyme because the enzyme evolved to bind to this transition state. That's how it works. And so since enzymes bind transition states with highest affinity, this strategy could potentially yield the best inhibitors. Binding to transition states is the same thing as saying you're going to catalyze the reaction. You're lowering the energy that's required for the reaction to take place. And here we're seeing a dramatic example of this. By binding to this transition state, you're forcing the substrate to get its carbons in the right location for this reaction to take place. And if you don't do this, the reaction, the stuff is just swinging all over the place. And if it's swinging all over the place, it's finding lots of other stuff to do with its time, and the reaction never takes place. I have so much more I want to tell you about. When we come back on Thursday, I have something really cool that I've been saving up for a while. I want to talk to you about how it is that enzymes work, not just in terms of binding transition states. by the way, this is kind of the classical stuff you learn in biochemistry. It's not wrong. It's actually totally right, or else I wouldn't say it to you. But I want to talk to you about the other aspect of enzymes, which is enzymes also have motion. They actually are going to be physically forcing together these transition states to lower the transition state energy and in turn accelerate reactions. And to me, that's really one of the most extraordinary aspects of enzymatic catalysis. So let's stop here. When we pick it up on Thursday, we'll be talking about motion as a mode for accelerating chemical reactions.